Muted.
Good morning, everybody. My name is Glenn. I'm the Antibody Marketing Specialist at GenScript. And today I've invited Dr. Woo-Sung Yin, the Associate Director of our Antibody Department at GenScript, to talk about premium hybridoma development and their application in antibody drug discovery. Just a brief introduction. Dr. Yin received his PhD from the University of Massachusetts Medical School, specializing in antigen presentation and CD4 positive T-cell epitope selection. He then joined Pfizer as a postdoctoral fellow, specializing in immunogenicity of therapeutic biologics. Dr. Yin has extensive experience in antibody drug discovery, especially with DNA immunization, high-throughput FACS screening, ELISA kit development, and anti-idiotype antibody development for therapeutic antibody drug PD and PK studies. In this webinar, he will first give an overview of antibody drug discovery and then talk about the Hybridoma platform at GenScript. After that, he will review our suite of antibody characterization tools and finish with some case studies involving therapeutic antibodies generated through hybridoma technology. We hope this will be highly informative, and please take this opportunity to ask Dr. Yin any questions that you may have regarding hybridoma development and its application in antibody drug discovery. To ensure the sound quality, all attendees are muted. Feel free to type in your questions in the chat box in the lower right corner of the screen. Dr. Yin will address your questions together at the end of the presentation. So without further ado, let's welcome Dr. Yin to give his talk on premium hybridoma development and applications in antibody drug discovery.
Thank you, Glenn, for the introduction, and thanks everyone for attending this webinar. As mentioned, I will talk about the hybridoma generation for therapeutic antibody drug discovery applications. So this is the outline of my presentation. I will first give an overview on antibody drug discovery, and then focus on platform and the technology of that. I will also discuss multiple antibody characterization assays during antibody drug discovery. Following that, I will give a comprehensive case study of identifying therapeutic leads against an immune checkpoint target using hybridoma approach, and I'm happy to answer your questions after that. So antibody drug discovery and the development process is really complicated, and here I list several essential steps. The process starts with the target assessment and validation based on literature reports or in-house tests. After a target-specific project plan is established, reagents and assays for screening of specific high-affinity and functional antibody will be prepared. Antibody hits generation will be followed with either library approach or hybridoma approach. After you obtain hundreds of thousands of hits from screening, you can further narrow down the list by performing favorable biochemical, biophysical, and functional assays for selection of leads. Leads optimization is yearly followed, including chimeric antibody generation, antibody humanization, and antibody affinity maturation. After that, large-scale antibody production will be performed for further toxicity and efficacy studies. Based on those data, a few candidate antibody molecules will be selected and further characterized with stability and developability assays. Final selected candidate will enter clinical trials and need to pass extensive regulatory agency evaluations before entering into the market for therapeutic indications. So this is the overall path for the antibody drug discovery process. There are several technologies to generate therapeutic antibodies. Hybridoma approach with human transgenic mice or B cells from immunized human body can directly generate human antibodies. The most popular one is still hybridoma approach with rodent system, in which you get rodent antibodies and then perform humanization to get humanized antibodies. Phage or yeast display is also an option to get humanized or human antibodies, depending on the library built. Besides the hybridoma and the library approaches, there are other technologies for generating therapeutic antibodies, including the SLAM technology, which recovers the variable region sequence from specific single B cell from immunized animals and is able to generate humanized antibodies. More recently, next generation of antibody sequencing from naive or immunized individuals can also be applied to generate human or humanized antibodies. So this is several cartoons showing how the hybridoma approach and the face display approach be used for antibody lead generation. As mentioned, hybridoma technology is traditional since 1970s, but still powerful. By 2015, FDA has approved more than 50 therapeutic antibody drugs, and 49 of them were derived from hybridoma technology, and only 3 from face display platform. The hybridoma technology still generates high market value by inputting more therapeutic antibodies into the market. Here are several approved antibody drugs from each technology platform. As you can see that most of them are derived from hybridoma approach as fully human antibodies from transgenic mice, colored red, or humanized antibodies from rodent system, colored blue. As you can see, most of them are from hybridoma. Next, I will discuss the hybridoma platform to generate the therapeutic antibody leads in antibody drug discovery. So this is the overview of hybridoma platform for antibody drug discovery. Hybridoma generation and lead identification is the start. After getting the antibody leads from hybridoma approach, antibody engineering is performed for lead optimization, including antibody humanization and affinity maturation. Usually, leads from hybridoma approach have reasonably high affinity with KD in nanomolar to sub-nanomolar range, and therefore antibody affinity maturation may not be necessary, especially with affinity guaranteed humanization process. During antibody drug discovery process, multiple in vitro and in vivo functional assays are needed to characterize and screen for functional antibody from numerous binders. After an optimized lead is obtained, transient or stable expression of lead antibody is performed for producing large amount of materials for further efficacy, toxicity, stability, and durability studies. Several critical paths for antibody lead identification by hybridoma approach. We start with reagent preparation, including immunogens, positive screening and negative screening materials, as well as assay reagents. Animal immunization is followed, and meanwhile assays for anti-serum titer test, hybridoma screening, and his characterization are being developed. So the next step is to select animals for fusion, based on titer test or other functional evaluation of the anti-serum from each immunized animal. An average of 20,000 to 50,000 hybridomas can be obtained, which will be subjected to several rounds of binding, ligand blocking, functional screening. After multiple hits are generated, subcloning is performed to achieve single stable monoclone for lead selection based on further biophysical and functional assessment. Lead optimization is such as humanization is often performed with affinity maturation when necessary. Next, the leads will be subjected to toxicity and efficacy evaluation in animal models for candidate selection. So conventional hybridoma generation procedures follow immunogen preparation, animal immunization, fusion and hybridoma screening, subcloning and hybridoma cryopreservation, and antibody production. GenScript has extensive experience in all of these critical paths, from multiple approaches on immunogen preparation to variety of choice on host animals. We also have high efficient electrofusion platform and high throughput screening platforms for identifying binders against the most native epitopes. Moreover, we have a very experienced team for project management, and in the last 10 years, we have successfully delivered more than 10,000 antibody projects, including over 100 therapeutic antibody projects. In the next couple slides, I will expand a little bit on each of our expertise in the critical path. So we have multiple choice of immunogens on a targeted basis, from peptide, which can be native or modified, to protein, including secreted proteins, extracellular domain or membrane proteins, and membrane preps. Whole cell and DNA immunization are suitable for receptors, including GPCRs, surface antigens, including immune checkpoint proteins, ion channels, and transporters. Virus-like particle is efficient in incorporating membrane targets for immunization. We can also use a combination of the various immunogen options following specific project requirements. As you can see, these are CHO-K1 cells overexpressing the target, and we can use gene gun to deliver the DNA plasmids for DNA immunization. So DNA immunization, also called gene immunization, is our special strategy to generate monoclonal antibodies against membrane-bound proteins, such as immune checkpoint proteins that purify extracellular domain protein may have altered conformation, or against GPCRs and ion channel proteins that are hard to express, purify, and prepare as immunogens. In DNA immunization, host animals are injected with plasmid encoding full-length target proteins. Target protein expressed in vivo can elicit the immune response, as you can see from the cartoon here. So in DNA immunization strategy, the immunogen can be the DNA plasmids only. But based on our experience, DNA plasmid priming followed with boost immunizations of cells overexpressing the target protein or extracellular domain of the target protein will enhance the anti-serum titers. Multiple species of animals are available for DNA immunization, including BAP-C and C57 mice, Vista rat, and transgenic gamma mice if high affinity antibodies are desired. The DNA plasmids are administered into the animals by gene gun system, and with 3 to 15 microgram DNA per animal, and 5 times to the 6 overexpressing cells, or 10 microgram protein for boost. So this is the overall strategy for DNA immunization option. In the case that our human target has high homology with the rodent counterparts, we have the proprietary ImmunoPlus technology to break the immune tolerance. In the case shown below, before applying this technology, all the 4 mice did not generate a sufficient antibody response against the high homology target. However, after applying the ImmunoPlus technology, all mice generated sufficient anti-serum titers, as you can see from the bottom right figure. Another application of this ImmunoPlus technology is to develop surrogate antibodies such as mouse anti-mouse antibody for target validation. So after the project is evaluated on a target basis and the immunogens have been prepared accordingly, we could start immunization and hybridoma fusion. As mentioned, multiple choice of animal strains are available, and in most cases they can reach a very high titer after third immunization with an average titer of 512 K. Compared with library approach, hybridoma approach offers a great advantage of high developability of antibody leads, but has the intrinsic disadvantage of missing potential antibody sequence due to low fusion efficiency. The electrofusion method we used guarantees high cell fusion efficiency to increase the size of hybridoma for screening. We observe an average fusion efficiency of 1 hybridoma over 2,000 B cells, resulting 20,000 to 50,000 hybridomas per animal. The plot on the right is a summary of the hybridoma obtained from our recent 575 projects. Compared with the traditional PEG fusion, which usually results in 10,000 to 1,000 to 2,000 hybridomas, the high efficient electrofusion generated an average of 50,000 hybridomas per screening. So in this way, the hybridoma approach preserves the good feature of the high developability of leads generated from this approach, and also with our high efficient fusion process, we can increase the size of hybridomas to be screened for therapeutic leads. After we obtain a large number of hybridomas, the next step is to perform hybridoma screening for binders. We have multiple natural epitope-based high-throughput screening platforms for soluble target. Both capture and indirect ELISA are in place. For membrane target, we have BD FAX caliber with high-throughput loader and high-throughput IQ screening system, which can read a 96 well plate in 3 to 5 minutes by FAX. We are also equipped with a homogeneous HT-match binding assay system for membrane target binder screening. With these high-throughput screening systems, we are able to identify numerous hits of binders. So after we get a large number of hits, multiple characterization methods need to be performed to identify highly specific, high-affinity functional antibody leads. In the next few slides, I will talk about the therapeutic antibody characterization, which is necessary to identify fit-for-purpose therapeutic leads from numerous binding hits. So before I go into the characterization assays, I want to bring your attention to the features of antibody drug discovery projects. On the left side is a general workflow for a regular monoclonal antibody project aiming to identify ELISA application to an antibody with a straightforward streamline from immunization to ELISA test of test bleed titers, and then select animals for fusion with screening by ELISA. Based on ELISA screening, subcloning is performed to obtain stable monoclones, and the last step would be ELISA QC or purified antibodies. Unlike regular MAB project, on the right side is the general workflow for a representative antibody drug discovery project for membrane target. As discussed, the goal of antibody drug discovery project is to identify specific high-affinity functional antibody. Besides ELISA test of bleed titers, FAX is also performed to check the membrane-bound antibodies, and often the test bleed will be further screened with client's in-house functional assay. After selecting animals for fusion, besides ELISA screening, FAX screening is also needed. Subnatants or parental clones are often provided to clients for in-house functional assay with reference information of those subnatants included, such as total IgG concentration, relative affinity by ELISA EC50, ligand blocking activity. With all this data together, certain clones will be selected for following subcloning. Subcloning will again be screened with those assays, as well as the final purified antibodies. As you can see from these two workflows, antibody drug discovery projects are way more complicated in terms of screening and characterization assays to identify therapeutic leads. So as mentioned, therapeutic antibody discovery aims to identify highly specific functional clones as the leads for downstream antibody drug development, and therefore multiple antibody characterizations are needed. A pack of characterization tools are available at GenScript, including binding affinity and relative affinity ranking by EC50 for selecting high affinity clones, ligand blocking activity by IC50. Target specificity and cross-species reactivity can also be evaluated for lead selection. ELISA or SPR-based epitope binning can evaluate whether the antibody hits bind to the same epitope or not, and definitive antibody affinity measurement can be performed by Bioco. Functional IC50 or EC50 can be determined in cell-based functional assays. In the next few slides, I will give you some examples and case study data for those characterizations. So in this study, We aim to measure the binding activity of purified antibodies to antigens by ELISA EC50 assay to select antibodies with the desired binding affinity for downstream functional assay. As you can see from the bottom of the plot, these 10 MABs showed distinguished binding activity, and the corresponding EC50 was calculated. Based on our experience, antibodies with EC50 in the range of nanogram per ml show decent binding. For membrane target, we can also use flow cytometry to evaluate the binding activity of purified antibodies to membrane-bound targets, as demonstrated in the bottom right plot. So this is EC50 to measure the binding activity of purified antibodies to antigens. For most of the targets with known antibody receptors or ligands, the functional clones usually have the feature of ligand blocking activity. Therefore, ligand blocking activity characterization is often for Hybridoma supernatant during screening or for purified antibodies or final clones. In this example, 10 MABs were tested for their ligand blocking activity, and as you can see from the data, that they show different blocking activities. MAB8 and MAB9 could not block the binding of ligand to target protein at all. From this data, antibodies with strong ligand blocking activity, such as MAB10 and MAB3, in this example, can be selected for further characterizations. For membrane targets, ligand blocking activity can also be evaluated by FACS. In this example, antibodies against PD-L1 were generated and the ligand blocking activity was assessed by FACS. As shown in the bottom left figure, a pilot experiment needs to be done to find out the appropriate ligand concentration, which is 0.2 microgram per ml of PD-1. Then the ligand blocking experiment was performed using that concentration of PD-1. As you can see from the bottom right, MAB1 showed strong ligand blocking activity, which is an antagonist antibody. MAB2 did not show any blocking effect. Interestingly, MAB3 showed ligand stimulating effect, which is agonist antibody. After the ligand blocking activity assessment, desired antibodies can be selected for further lead characterizations. Another characterization for lead selection is to measure the cross-species reactivity. In most cases, cross-reactive antibodies are desired for future animal studies. In this example, MAB1 and MAB2 generated against human targets were tested for their cross-reactivity against human, mouse, rat, and monkey targets. As you can see from the bottom left figure, MAB1 showed binding reactivity to human target as expected, but no binding to mouse and rat targets. MAB1 also showed cross-reactivity to monkey targets due to high homology between monkey and human targets. MAB2 also showed good binding to human and monkey targets just like MAB1, but also decent although lower binding to rat targets, and no binding to mouse target at all. With this cross-reactivity data, if you want to use monkey for animal studies, you can use either MAB1 or MAB2. But if you want to use rat for animal studies, only MAB2 is suitable. Neither MAB1 nor MAB2 works in mouse models. In that case, you need to develop or surrogate a mouse anti-mouse antibody for target validation in mouse models using our ImmunoPlus technology. Epitope Binning is another useful characterization tool for lead selection, in which antibodies are grouped by their binding epitopes on the antigen. Antibodies bind to different epitopes may have distinguished binding modes, mechanism of efficacy and safety concerns, as well as antibody-drug developability. In this case study, competitive ELISA was explored to group epitopes of 4 antibodies in a checkerboard format. As you can see from the data, unlabeled MAB1 could compete the binding with the built-in labeled MAB1, which is expected as the self-validation. MAB2 and MAB3 can also efficiently compete the binding with MAB1, which shows that they probably bind to the same epitope. In contrast, MAB4 could not compete the binding against MAB1, but could compete with itself. Taken together, this data demonstrate that MAB1, MAB2, and MAB3 bind to the same epitope, while MAB4 binds to a different epitope. Besides ELISA, epitope binning can also be performed by Buccal-based assay, as shown on the right. If 2 antibodies bind to different epitopes on the antigen, an additional signal will be observed. Select antibodies in different epitope groups for further lead evaluation, or you can select antibodies share the same epitope with non-antibody candidates. So using the Hybridoma platforms and our over 10 years expertise, we have successfully raised antibodies to various targets, including soluble targets like cytokines, single transmembrane proteins like the immune checkpoint proteins, multiple transmembrane targets like GPCRs and ion channels. In the recent years, therapeutic antibodies targeting immune checkpoint proteins have achieved great success in multiple oncology indications, including Yervoy against CTLA-4, Keytruda and Opdivo against PD-1, and Tecentriq against PD-L1. We catch the wave and accumulate extensive experience in identifying therapeutic antibody leads against those targets. So in the last few slides, I will show you a case study of an anti-immune checkpoint antibody lead identification through Hybridoma approach. This is the overview of this comprehensive case study. In this practice, 10 mice were immunized and the test bleed were evaluated by both ELISA and FACS. After that, 2 animals with top titers were performed for fusion, and the generated hybridomas were plated into 6596 well plates. After primary and confirmative screening, 843 ELISA positive wells or parent clones were identified, and 86 of them were confirmed with ligand blocking activity by ELISA, of which 26 parent clones were confirmed with binding by FACS, and they proceeded to 1-round subcloning. 53 1-round subclones were obtained, and we selected 28 of them for antibody microproduction. These 28 antibodies were validated by mixed lymphocyte reaction functional assay, which is a standard functional assay for immune checkpoint antibody lead identification. After the single dose mixed lymphocyte reaction functional assay, 12 of the 1-round subclones were further subcloned to obtain stabilized final clones, and then larger scale antibody production was performed. For those 12 antibodies, multiple characterizations, including ELISA EC50 and FACS for binding activity, ELISA IC50 and FACS for ligand blocking activity. After those characterizations, 5 antibodies were selected for multi-dose mixed lymphocyte reaction functional assay and epitope binning. In this way, we showed how functional leads were identified through Hybridoma approach. In the next few slides, detailed data will be presented. This slide shows the ELISA EC50 to evaluate the binding activity of 12 antibodies using the marketed drug as the reference. As you can see that Many good binders have been obtained with comparable or even better binding activity compared with marketed drug. As you can see, marketed drug has an EC50 of 7.3 nanogram per ml, and we have several clones with lower EC50, which means better binding activity. In this slide, the 12 antibodies were evaluated for their ligand blocking activity. And again, we have obtained several antibody leads comparable to marketed drug. There are several antibodies showed very low ligand block activity, such as 21H11D7 and 23F12E2A9, which probably should not be selected as leads for further downstream development. This slide evaluated the surface binding of 12 antibodies to cell lines overexpressed. As you can see from the data, all 12 antibodies showed decent binding to membrane-bound targets. In this slide, ligand blocking activity of the 12 antibodies was evaluated by FACS against membrane-bound target. Most antibodies showed efficient ligand blocking activity, but not 21H11D7 and 23F12E2A9, which is consistent with the ELISA IC50 results. After those binding and ligand blocking activity tests, 5 antibodies were selected for multi-dose functional assay, which is the mixed lymphocyte reaction assay. Again, the marketed drug was used as a reference control, and we have obtained therapeutic leads with comparable in vitro functional activity with the reference marketed drug, as you can see from the dose-dependent curve. In the last slide of the case study, Epitope Binning was performed for the 5 antibodies to see whether they share the same epitope or not. As you can see from the checkboard assay format, 4 antibodies could compete with the marketed drug for the binding of antigen, which showed that probably they share the same epitope. In contrast, the antibody 23F12E2A9 did not compete with other 4 antibodies as well as the marketed drug, which showed that very likely this antibody binds to a different epitope on the antigen. If you remember from the previous ELISA IC50 and FACS blocking data, this antibody also did not have ligand blocking activity, and this is consistent with the idea that it binds to a different epitope. Another message is that 1H10C4D4 and 16H5D5B4 may have stronger affinity than marketed drug to same epitope on the target, as demonstrated by better competing ability with marketed drug, which is also consistent with previous ELISA EC50 data. So far, we have shared a case study of how therapeutic antibody leads were identified by Hybridoma approach and how the characterizations are utilized to narrow down the leads to find the desired leads for future downstream development. Here is a quick summary for advantage of GenScript antibody drug discovery platform, including multiple immunogen and host animal choice and optimized adjuvant and immunization schedule, as well as our ImmunoPlus technology to break the immune tolerance for high homology targets and for surrogate antibody development. Our high-throughput screening platform enable us to efficiently screen numerous hybridomas and obtain multiple hits. Multiple characterization tools are available to narrow down the hit leads to identify specific high-affinity leads. In vitro and in vivo functional assays are in place for functional lead selection. Downstream assay development such as sandwich ELISA, using pairing antibody, and PK and immunogenicity evaluation using anti-elotype antibody. Lead optimization such as humanization and affinity maturation, as well as large-scale antibody production are also our expertise. In the last couple slides, I will give you an overview of our facility and overall antibody response service. GenScript has over 12 years experience in antibody production. Our animal facility is ALAC and OLAW double accredited, hosting more than 8,000 rabbits and 12,000 rodents, and providing a broad range of antibody service from individual cases to bulk orders. We are equipped with high production capacity and can produce 500 to 600 monoclonal antibody and 50 to 80 monoclonal antibody projects each month. If you have any antibody production need, GenScript is a one-stop shopping place for you. This is an overview of the antibody service we offer at GenScript. Besides the antibody drug discovery related service I just introduced, we also have the anti-ideal type antibody generation, surrogate antibody, and the immunogenicity assays. We also offer reagent antibody service for polyclonal and monoclonal phosphor-specific antibodies, neutralization and blocking antibodies, as well as antibody pairing. We also offer more than 600 catalog antibody products, diagnostic antibodies, and immunoassay development, such as ELISA kits for endogenous protein detection, host cell protein detection, PK, and immunogenicity evaluation. So thanks for your attention, and I hope you have learned something useful for your antibody drug discovery projects. With that, I will pass the ball to Glenn for the Q&A session, and I'm more than happy to answer your questions. So thank you.
Thank you, Dr. Yin. Very informative. Appreciate that. And thank everybody for attending this webinar. So we'll move on to Q&A now and answer some questions for you as they roll in. So looks like the first question, just in general, Dr. Yin, how long would you say ballpark the timeline is for antibody drug discovery?
Okay, so this really depends on the target, because antibody drug discovery is highly customized on a target basis. But in general, the antibody lead identification takes about 6 to 12 months, and the lead optimization takes another 6 months.
So just to clarify, cumulatively that could be a year to a year and a half for the entire project?
Yes, exactly. But different targets has different timeline, but this is general, that's timeline.
Understood. Okay, so specific to the DNA immunization, how does that compare as far as success rate? You know, what is, it's sort of new, so what is the success rate for DNA immunization?
For a DNA immunization that we have used for over 6 years, for single transmembrane targets such as immune checkpoint proteins, our success rate of DNA immunization is over 95%. For multiple transmembrane targets such as GPCRs and ion channels, due to the extremely complexity of the targets, our success rate of DNA immunization is about 40 to 50%. Which is still high in the field, even though it's relatively low compared with single transmembrane targets.
And just to follow up on that, so the success rate, although it potentially looks low, is 50%. That's far superior to non-DNA immunization, correct? For something like a high homology target.
Yes, because especially if we combine with the ImmunoPlus technology, because we can for DNA immunization. We can also apply that technology by introducing extra modulator in the plasmids used for immunization.
Gotcha. So both technologies can be used simultaneously. Excellent.
Yes.
Okay. And you gave a recap, but if you could just describe how antibody drug discovery is different from regular antibody discovery when they're used for reagents.
Yeah, that's really a good question, because for regular reagent antibody discovery, The screening process is much easier because you identify a single application, such as ELISA binder or WB application antibody. So that's the first point. The screening process for reagent antibody development is much easier. So second point is the QC standard of reagent antibody development is much lower because it's a reagent, and you imagine. And for antibody drug discovery, you need all those characterizations from affinity, specificity, and function from all different ways to characterize functional leads. So that's the second point, the QC standard. So thirdly, that the documentation requirement of reagent antibody development is much lower because for antibody drug discovery, you have to prepare the full lab notebooks and all other documents to show that you have the capacity and all the instruments used are confirmed and all the material used are confirmed. So the documentation requirement for antibody drug discovery is much higher. And the last point is the need for high throughput platforms or reagent antibody development is much lower because for reagent antibody, you just need several binders. For antibody drug discovery, you have to screen the functional clones from the multiple binders. So you have to enlarge your screening pools, and in other words, you need a high throughput platforms. So basically, those are those 4 points that show the difference between the antibody drug discovery project and a regular antibody project.
Excellent considerations to take. Thank you. I think we have 2 more questions. Here's another one. In general, which is the most time-consuming step in antibody drug discovery?
So the most time-consuming step is the functional assays, I have to say, because for some assays, for some targets, the assays are very easy to explore. For example, for some of those cytokines, we just use cell proliferation assays. For some of the targets, such as immune checkpoint targets, such as LAG3, TIM3, the functional assays are very hard to establish because you need to get the PBMC from the blood to establish that assay because those targets are in the immune system. So I have to say, in general, the most time-consuming step is to establish the functional assay. And for the others, because we are very experienced, we can estimate the timeline from the immunization to the fusion screening and subcloning and antibody production. Those can be controlled and very well controlled. But the functional assays is complicated.
Understood. So that variability that you first mentioned, the 6 to 12 months, mostly comes from the laying the groundwork for validation for application-specific or development of your quality control for the functional assays. That's the variability.
Yes, exactly.
Understood. So obviously that's something you would want to have laid down first before you enter into it to make sure you could, screen appropriately as your immunizations come online, correct?
Yes, exactly. So we have to, as I mentioned in the critical path, so before we start immunization, the materials and the screening assays has to be established.
Excellent, excellent. Okay, here's an interesting one. What would you say are the advantages or disadvantages of using transgenic mice for development of full human antibody as therapeutic leads? So this is, I guess, looking forward into direct translation of the antibody, and if you can give your opinion on that, I guess.
Yes, yeah, sure. That's a very good question. The advantage of the transgenic mice for development of fully human antibody is that using this platform, there is no need for time-consuming humanization process because usually humanization it takes about 6 months. The other advantage is that the developability of leads from this platform is much better because those leads are nature. You don't have to do any engineering. So those are nature. So the developability is much better. But there are 2 obvious disadvantages. The first one is the cost of early stage is much higher because you have to pay for the technology access fee, and you have to pay for the mice, the transgenic mice fee, and you have to pay annual fee, and you also have to pay the milestone fee and loyalty fee. So the cost is very high. And the second disadvantage is the access to this platform is very limited. Only very few companies can offer this service, and most of the companies they are purchased by big farmers. So not very few are available in the market to offer the transgenic mice for development of fully human antibody.
Right, so it comes down to a balance between cost and time and access, I guess, as well. So
yes, exactly.
Good considerations. All right, we have one more as of now. So what rationales or limitations would you suggest, or do we have in choosing what type of immunogen, specifically asking between peptides, proteins, cell lines, DNA, and I might add to that question a combination of those. How would we, view those individually, and then as a collective, how would you use those as an immunization approach?
Yes, sure. So for peptide immunogen is cost effective, so it's very cheap, and it can specify targeting epitopes. So that's a good thing. You know where the antibody are targeted, but may not have correct or native conformations. So that's a big concern. Protein immunogen is usually expensive, especially for mammalian expressed, and may have the concerns of incorrect or non-native conformation, but they are very immunogenic. Usually we have very high titers if you use protein immunogens. So cell line immunogen has native conformation, but it usually have relatively low abundant immunogen because there are a lot of other non-specific protein on the cell surface. And the good thing is that it has the native conformation of the target protein. So that's for cell line immunogen. For DNA immunization, it can bring native conformation of immunogens and it can last longer in vivo. The drawback is the immune response is usually low and the cost is relatively high, and the immunization timeline is relatively long. That's why in general, on different targets, we will design different immunization strategy. For example, for cytokine, for soluble target, we just use protein immunogen because it's very immunogenic, and usually we don't have the concern of the non-native conformation. For single transmembrane protein, we can use either extracellular domain of the target, or we can use the DNA immunization plus boost with the extracellular domain, or DNA priming and boost with the cell line, or expressing the target. For some multiple transmembrane targets, such as GPCR, usually we have to use the peptide as an immunogen because it's very hard to generate the GPCR protein. And usually we use the DNA and priming and plus the peptides, extracellular peptides for boost. Or we can use DNA priming and the cell lines overexpressing the GPCR for boost.
Excellent. So, there's actually a follow-up to that question a little bit. One we didn't include actually as far as immunization. What are your views on using the phage display platforms for screening for antibody discovery compared to an animal-based immunogenic method? So how would you compare those two approaches?
So phage display approach has a clear advantage that it has huge library. It basically can cover all possible antibody combination, way region and way light chain and heavy chain combinations. So in general, you won't miss anything. But the drawback is that for phage display approach, the developability of those antibodies, especially sometimes affinity, are low. So you have to do further engineering, such as the affinity maturation. For the Hybridoma approach, as I mentioned, it has a good advantage of the good developability because it's nature. All the antibodies are developed nature, so it has a high affinity. Usually, you don't have to do the affinity maturation; you just have to do the immunization, and so the developability is much higher. But has the intrinsic limitation is that hybridoma approach, you can only fuse some B cells, and potentially you will miss some good candidates. So usually some clients, they use both strategies in parallel to maximize the success rate. But most of the clients, they choose a hybridoma approach because it has a better developability, and also most of the antibody drugs approved are developed by hybridoma approach.
Interesting. So the perspective to simplify that essentially is phage display will get you all your hits, but then you have to put in time and effort to generate the appropriate affinity, whereas a animal-based hybridoma will give you the affinity, but you may have to do multiple parallel hybridomas because of fusion efficiency.
Exactly. Yes.
If I could follow that up, just one more to clarify, what's your perspective as far as the combination of those with a immunized library rather than a naive library, if you were to start from that for phage display?
Yeah, immunized library in principle you should have a much higher hit rate, but still these are engineered. So all engineered because you just make all those potential combinations, and in some cases you may have a native combination, but still in a lot of chance you have engineered pairing.
Understood. So you're still possibly looking down the developability issue in the future. If you get all more positive leads, less rounds of phage display and panning, you'll still have to deal with affinity. Okay. If there are no more questions, I'd like to thank everybody for attending this webinar, and thank you, Dr. Yin.
Okay, thank you everyone, and thank you, Glenn. Thank you. Bye bye.
Thank you.